So in the previous slides, we took a look at how different kinds of hormones can affect a cell and cause that cell to change. But this slide relates to what in the world causes an endocrine gland to secrete its product, its hormone, in the first place. There are three basic mechanisms or stimuli that can cause an endocrine gland to change what it's doing. Now, we use the word stimulus, but in fact, changes in these three things, humoral, neural, and hormonal, can inhibit an endocrine organ. And we're going to look at those mechanisms more in the next two parts of the chapter. So let's take a look at these three mechanisms. Humoral stimuli. This is when the change in the chemistry in the blood will trigger a change in what a gland secretes. So again, the hormone release or inhibition is caused by changes in the level of ions and nutrients in the plasma of blood. This humoral stimulus, this part A, shows the effect of changes in blood calcium. If it becomes too low, that is detected by receptors in the parathyroid glands and causes the parathyroid gland to secrete its major hormone, which is parathyroid hormone. And that in turn will increase blood calcium. That's a really nice negative feedback cycle, if you'll recall from A&PII 1. When we take a look at B, or the middle of this slide, the neural stimulus, this is an example of how neural signals from the nervous system to an endocrine or neuroendocrine tissue will change its behavior and what it secretes. And so again, if you studied the autonomic nervous system, this should be familiar, that a signal from the central nervous system through the sympathetic pathway to the adrenal medulla, and that adrenal medulla is modified neural tissue that will secrete a neurohormone called mostly epinephrine, but also a little norepinephrine. And then those products are secreted into the bloodstream. So neural stimulus to a neuroendocrine gland, like the adrenal gland, the adrenal medulla. Now looking at the third part of the slide, C, the hormonal stimulus. This is when one hormone that is released will cause the release of a second hormone. And we call this a tropic effect, or you may see it spelled trophic, P-H-I-C. So the word is tropic, not tropic. We're not going to the Bahamas on this trip. But this illustration here is something we will again look at in more detail later in this chapter. And that is the effect of the hypothalamus, which secretes regulatory hormones. And those regulatory hormones target the anterior pituitary. That means the regulatory hormones are tropic because they cause the anterior pituitary to secrete more or less of other hormones. So the anterior pituitary hormones can target the thyroid gland, Others will target the adrenal cortex. Still others can target the gonads, for example, the testes. That means the anterior pituitary hormones are tropic because they are targeting other endocrine glands. Now, it is possible then for example, the testis can influence the secretion of other hormones, and we will look at that actually in the last chapter of the semester. The previous slide, the one before this one, talks about what causes a hormone to be released in the first place. This slide is all about how that hormone is going to cause changes in what the cell does. In other words, what makes a target cell a target for a specific ligand or hormone? Well, the key word here is affinity. The chemical structure of a particular hormone is attracted to the chemical structure of a particular receptor, and that's what draws them together. They have an affinity for each other. Now, this process demonstrates something called specificity. If you flood a particular cell with hormone A, but they only have receptors for hormone B, hormone A will have no effect on that cell because there isn't a receptor for it. So it's very specific. A couple of other characteristics we need to talk about: upregulation and downregulation. This is a fascinating mechanism that still astounds me when I think about it. So if a hormone is being suddenly released in a large quantity, the target cells somehow detect that and insert more receptors into the cell membrane. Upregulation. Upregulation will make the cell more sensitive to that hormone because it has more receptors. Now, if you continue to flood that cell with that hormone, at some point the cell is going to say, yeah, well, the cell doesn't talk, but you know what I mean. The cell's response is, you have flooded our receptors enough. We're not going to respond anymore. And at that point in time, it actually pulls receptors out of the cell membrane. Is that not cool? That's just really cool. Anyway, that is called downregulation, and it makes the cell less sensitive to the hormone because it has fewer receptors. It's really cool. The other three terms on this slide relate to how hormones interact with each other. The term permissiveness applies to a situation where one hormone has to be present in order for another hormone to work effectively. At the very end of this semester, we look at the reproductive system, and we will see a great example of permissiveness relating to how testosterone can function more effectively in the testes. One hormone gives permission for another hormone to work more effectively. Another term, synergism. If you are working synergistically with someone, you're working to where the result is going to be greater than what you could produce on your own. In other words, the total is greater than the sum of the parts. Synergism. I'll give you an example of that in class. Another term, antagonism. Sorry about the misspelling. Something that is antagonistic is, in essence, opposite. So we do have situations where one hormone functions antagonistically with another, and we will see an example of that as we go forward. How long will any given hormone last? How do they get from one place to another? This is what's addressed by this slide. Your textbook talks about it. It's not in your notes, but I think it's important to address. In terms of transport, hormones can be transported as free hormones, or they have to be bound to a protein. So now I want you to think, which one of these could be transported as a free hormone, and which one would have to be attached to a protein? I'll answer that at the end of the slide, but think about it. Now, another thing that is going to influence how effective a hormone is going to be the concentration of that hormone in its active form in the bloodstream as it goes to its target organ. Now, the concentration is affected by the rate of a release. If it's released at a faster rate, the concentration will be higher. The other thing that has an effect on concentration is its half-life. The half-life of any chemical is the rate at which it takes it to degrade 50% of what started. In other words, how long, if you have 1 gram of a substance and it takes 20 minutes to end up with a half gram of a substance, then we would say its half-life is 10 minutes. It took 10 minutes to break it down to half of its original amount. Now, duration. How long does any given hormone have its effect? Well, that's going to vary from hormone to hormone. Some hormones only have their effect for less than a second, and other hormones can have their effect for hours and maybe days. What makes the difference is the rate at which they are chemically broken down, the rate at which they are degraded. All right, you ready for the answer to your thinking question? The answer is lipids must be protein bound. Why? Well, if they were free, they could just diffuse everywhere, right? They could cross all kinds of membranes. So to keep them in the bloodstream, in their inactive form, until they can be delivered to their target organ, their target cells, they have to be connected to a protein. And that's in contrast with our amino acid-based hormones. They can just float around free because they cannot easily cross any of the membranes without help. You may have come across the idea of signal modulation in A&PII, because quite frankly, we do not have 100% pure response from any given ligand at any given time. Most of the time, they are adjusted by other ligands. So here on this slide, we see that we have our primary ligand is going to activate a receptor, and an agonist, in other words, a chemical that does something similar, also activates the receptor, and you have an increased response. Another way that a signal can be modulated is if an antagonist will block the receptor activity, and then you'll either have diminished response or no response at all. Here's a great illustration of signal amplification. And I bring this up now to reinforce something you already looked at in this chapter. And I'm going to ask you again, what is amplification and what type of hormone would trigger this kind of response. And what could we call it? Is it direct gene activation? Is it second messenger? One more slide from the other text to help illustrate the fact that there are two different kinds of receptors in anatomy and physiology. Now, specialized cells or structures that can convert one type of energy to another. For example, here we have in your ears, as you learned in A&PII, you have vibrations that can be converted to an electrical signal through your cochlear nerve back to your brain. So that's one kind of receptor. It is a sensor. But the kind of receptor we are talking about here is a receptor protein that is either embedded in the cell membrane or found inside the cell. So just keep that clear. A lot of times I make the distinction between a chemical or ligand receptor and a sensory receptor.